Okay, people, welcome. So, in the last class we saw this, the definition of the reactive species is nebulous and it is more of an understanding. There is a wide range 10 orders of magnitude. நான் போகும்போது போட்டுக்கிறேன். There is a 10 orders of magnitude range or 11 orders of magnitude range could be in the second order rate constants that involve reactive species reactions. So, this is more of an understanding, no clear cut definition. So, this is one, and then we looked at some related aspects, atomic orbitals, molecular orbitals and so on and so forth, and some examples of reactive species. Then we started looking at some history of oxygen from the time the earth came into existence, and then we looked at some interesting data. Remember, this is 600 million years ago, this is present day. Okay. We came into existence 5 million years ago, so we are very close to here, and this is oxygen. It has gone up to about 35%, and the carbon dioxide was as high as 0.5% about 550 million years ago and then it came down now 0.03, 0.04% and so on. Okay. So, fine, this is where we stopped. So, let us continue from here. Okay. The other surprising information is that oxygen is more soluble in organic solvents than in water. Okay. Most people do not realize this, and because of this it can easily enter the cell through the lipid bilayer. Okay. And of course, oxygen is toxic, oxygen is reactive and therefore, it is toxic, and it can easily destroy the cells, it can easily get through the cell membrane. Okay. Keep this in mind, when you encase something in plastic including foods, you are not keeping oxygen out. In fact, oxygen very easily gets through plastic. Okay. The permeability of oxygen through typical plastic is high. Okay. Typically, when some people do experiments under anaerobic conditions, they cover their setups with acrylic, which is a plastic. Okay. Be very careful. Okay. If you buy standard equipment, standard anaerobic setups, they come with a certain pouch that you keep inside, which will kind of extract out all oxygen. There is a highly reactive compound there, to which the oxygen can react, and thereby that reduces the oxygen content. The plastic does nothing to the oxygen. Okay. There is a certain rate at which it goes through. Depending on the thickness of the material, the permeability of the material to oxygen will be different, and so you will have oxygen going through at different rates and so on and so forth. That is always there, but plastic is highly permeable to oxygen. Okay. If you want to keep oxygen out, use aluminum or something like that, much less. Then there will always be some level of permeability, but that is far less. Okay. In fact, some years ago we got interested in this, and I realized that people had not reported this, and they were talking about acrylic sheets for anaerobic things when we wanted to study hypoxia and so on and so forth, and Sonal wanted to study hypoxia. We are looking at acrylic sheets, and then I said, how can you use acrylic, because oxygen will go through acrylic and there is no way that you can maintain anaerobic conditions. Then we went ahead, and Sonal and Prerna, I think, got together, did some calculations as to the permeabilities and so on and so forth. We were going to write up something saying that this is happening, how do you report anaerobic conditions under an acrylic sheet, acrylic enclosure. And then something struck me, people have been accepting this for such a long time, therefore, probably we are missing something. So, I went and studied materials and methods and so on and so forth. There in one of the papers it was given these standard setups come with an oxygen reactive compound in a pouch, and that pouch needs to be kept inside the acrylic chamber, and that is what ensures the anaerobic condition, not the acrylic sheet alone. Okay. So, be a little careful. And then we of course dropped the idea of the paper, but the calculations were nice. We have, if you ask Sonal, she will tell you the numbers and so on and so forth. And we have already seen that reactive oxygen species that are formed from oxygen, They oxidize essential cellular components, all the biomolecules, all the 4 fundamental biomolecules. Okay. And anaerobic metabolism is dependent on the components being in the reduced state, and once it oxidizes it, that is completely gone. This is in the context of anaerobic growth. So, historically anaerobes were there before oxygen came into existence on the earth's atmosphere. When oxygen levels started rising, the anaerobes either retreated to anaerobic environments, okay, under the sea, much under the sea, deep under rocks and so on and so forth, where deep under soil, where there could be anaerobic environments, or evolved antioxidant defenses. Okay, that is when they started evolving antioxidant defenses, of course with the natural selection, those that did not have perished and so on and so forth. And the ones that evolved antioxidant defenses became aerobes, the ones that could not became extinct. Okay. So, this is pretty much gives you an idea of a historical perspective of oxygen in our Earth's atmosphere. Okay. In this context, Ankita had somebody asked about the oxygen levels going up in the last slide, right? Why did it go up and so on and so forth, right? Ankita has an answer. I have asked her to check the believability of it, the veracity of it. She still needs to check the source. Most likely it should be okay. If it is a textbook, if it is a published paper and so on and so forth, that is fine. If it is a blog, especially graduate students blog, graduate students are typically very excited. So, they do not make a difference between what is proved and what may be, and what is an extension that is created by in their own minds and so on and so forth. All this maturity happens when somebody goes through a PhD process, and that is one of the important things that they develop. Very clearly, there is a huge difference between writing is in your paper and writing maybe in your paper. Okay. A huge amount of work that goes on between a maybe and a is. Okay. Only few maybes can get converted into an is in the course of a thesis, a PhD thesis of about 4-5 years. Okay. So, that is a very important aspect. In science, we make a huge difference between those two. So, be a little careful about all these things, and people come to us because we know the difference between is and a maybe. Okay. So, yeah, there are speculations. It's perfectly fine to speculate. It's perfectly fine to draw inferences based on what you have. This exists, this exists, this exists, and therefore it is reasonable to say that this exists. Still fine, but if you miss out the fact they say it is reasonably fine to say this exists, then you take that for granted. No, that has not been proved. That has been speculated. Speculation is fine. You take it on its weight. Don't give too much importance to it. But if somebody has proved something, yeah, fine, then that is not a problem. Okay. And typically the student blogs tend to be mostly maybes, because they have not figured out the difference between an is and a maybe, and so on. So, you want to tell that to the class what you found out, and she will of course go back and figure out whether it makes sense, it certainly makes sense, whether it has evidence or not, she will figure out. Okay. Would you like to share with the class as to what you told me just now? Might want to come close to this so that at least it gets recorded here, or you want to take this and talk about it. Come. Yeah. You might want to turn that side so that you get captured. When you stand here, you will get captured also. Yeah. உங்களுக்கு தெரியுறாங்களா? This should be. இவங்க? Go ahead.
Yeah, like before the Carbonaceous period, so there were certain the tectonic plates, they came close towards each other, and during that time the land submerged and oceans and all they got formed. So the mineral nutrients and all they were present in the water bodies and oceans and all. So the cyanobacteria, they got lots of inorganic materials, the nutrients to grow, proliferate at a faster rate, and as a result there was a lot of oxygen that was produced into the atmosphere, and oxygen level increased to a great levels. But after that period there were certain forest fires, and there was volcanic eruptions, and then followed by forest fires and all, which led to destruction of forests, and as a result the carbon dioxide oxygen level decreased and carbon dioxide level increased into the atmosphere.
Thank you. You can leave it there. That I think will be. இது ஆஃப் பண்ணுமா அது ஒன்று போடுங்க நாங்கள் மாற்றிக்கிறோம் அது இல்லாட்டி பட் இன்டர்ஃபியரன்ஸ் இருக்கும் இல்லையா
இல்லை இருக்காது இது நீங்களே வச்சுக்கலாம்
தேங்க்யூ ஆனந்த் இது ரெட் பட்டனா இருக்கட்டும் வேணுங்கிறப்ப பண்ணிக்கிறோம். Okay, so that gives you some idea. You can all go back and check and so on and so forth. She'll of course check. Okay, let's move forward with our discussion. The toxicity of anaerobes to oxygen is actually used for therapy purposes also. Okay, I wish a doctor was here. So a lot of this course would be oriented towards medicine. Gangrene, which is the death of the tissue due to lack of blood circulation, that is actually gangrene. It can be caused by a bacterium, a Clostridium, right, which happens to be a strict anaerobe. So Clostridium infections have been linked to gangrene. I mean, rather now it is accepted, fully accepted that okay. So just to repeat, we lost audio there. So the anaerobe Clostridium causes gangrene, which is the death of tissue due to lack of blood circulation, and that can be partly treated with hyperbaric oxygen therapy. As we all know, oxygen can affect the aerobes also. Okay, 21% oxygen is actually toxic to aerobes. The oxygen concentration in the atmosphere is actually toxic. Okay, although even though they have antioxidant defense mechanisms, it can be toxic. Just keep this in mind, and so do not push it to the extremes, as we will just talk about it. Some data again from your book. The original source is a paper by Michaels. This is the effect of reactive oxygen species on cell survival when treated with radiation. Okay, when exposed to radiation, nuclear radiation, what is the effect of different extents of oxygen, and therefore different extents of reactive oxygen species on cell survival. This is the surviving fraction in a log scale. This is the dosage in gray. You all know that gray is a unit of radiation. When it is grown in nitrogen, in this case there is no oxygen at all. I guess they took care of the growth and so on and so forth. These experiments were done after that. So as they increase the dosage of radiation, of course the surviving fraction came down, but that is the highest. Okay. Even at 0.05% oxygen, there is a drop in the surviving fraction. At any dosage, if you take 20 16, let us say, this is about 3 into 10 power minus 3, whereas this is about 10 power minus 2. Okay. So there is a huge difference in the effect of reactive oxygen species on cell survival. So, 0.05% oxygen, 0.11% oxygen, 0.21% oxygen, 0.21, okay, not air. This is 0.44, 1%, 4%, 10.5%, and this is air, 21% oxygen. So, this can be quite drastic, the effect can be quite drastic on cells, very clearly shows the reactive oxygen species damage that it can do. Of course, this is shown as its ability to take in a radiation, but this gives you an idea as to the toxic effects of reactive oxygen species. Okay. So, the main message here is watch out for oxygen, right. Although high pressure oxygen is used to treat nitrogen bends and so on and so forth due to rapid surfacing when divers come up quickly, they can get bubbles entrapped in their vasculature, and that causes tremendous pain, called bends, nitrogen bends. And people have used hyperbaric oxygen therapy to treat that, that is fine. Whereas pure oxygen and oxygen supplementation is used for resuscitation of newborns to improve oxygenation of the fetus, that is delivered by cesarean section. It is done in a very standard fashion. Okay. All these are done. Okay. COVID situation, you all know that you had to pump in oxygen, and that was a major problem in the second phase of COVID and so on and so forth, which led to a lot of deaths. Okay. So, definitely when it is a choice between two devils, as it usually is in the case of allopathic treatments, whenever in some allopathic treatments, then you will have to make a rational choice. Okay. I am reminded of the training, the safety training that I went through, where I guess when you take up certain positions, you will have to go through trainings and so on and so forth. So, we were given some training on first aid, and that included CPR. So, the person demonstrated CPR and so on and so forth. So, I was quite curious, the way the person was doing it on a dummy was quite vigorous. So, I said, what happens if you break a bone, especially on top of your lung? If you break a bone, that it can puncture the lung, and that could be disastrous. But I said, yeah, that could always happen. However, that is lung, bone damage, probable lung damage. You can handle it later. Okay. Here, if you do not do that, the person is gone. Right. So, it is a choice between two devils, the smaller devil is chosen, and that is how you need to view these situations. It is not that oxygen is nice and all that, it is also toxic, but in this particular situation you need that level of oxygen. It can cause acute nervous system toxicity resulting in convulsions within 6 hours of exposure. That is what this one says. Just 1 atmosphere pure oxygen for 6 hours can cause chest soreness, cough, sore throat. 24 hours exposure results in alveoli damage, right, the bottom part of your lung vasculature, which is essential for gas exchange and all that, alveoli damage, and consequent fibrosis, which is laying down of inelastic fibrous material in the lung, which permanently impairs gas exchange. Okay. So, this is known with regular 21% oxygen. Okay. This is pure oxygen, not 21%. This is pure oxygen, and therefore, also in premature babies who have not yet developed antioxidant defense systems, it takes some time for their immunity to develop and so on. Even 21% oxygen can cause something called retinopathy of prematurity, which is formation of fibrous tissue behind the lens, which leads to blindness straight away. Lens is gone. Okay. Nothing, you cannot do anything about the fibrous tissue behind the lens, so that is gone. So, this is a known problem. So, you will have to watch out for all these things. All this is because of the reactive oxygen species. Therefore, watch out for those oxygen therapies, which are quite popular, especially in Japan. After a long day's work, people used to go to oxygen bars and inhale pure oxygen. It was a fad here too some years ago. Be a little careful, it can cause significant damage. Any questions, comments? Yeah, we might as well start discussing now and rather than at the end. That way we'll have some time. Yeah.
In premature babies, even 21% oxygen causes retinopathy prematurity, but why does it occurs only in the lens, not in the other tissues? Oxygen gets exposed even through nasal as well.
It could happen in many places. There is nothing that prevents the fibrous tissue formation could be dependent on so many other things, right. Maybe that happens much more quickly in the eye. I am guessing, I do not know, I am guessing, but based on fundamentals I would say that it can happen anywhere, not the rather the damage can happen anywhere. The cause of fibrous tissue which leads to permanent damage probably is more observable in the eye, I would guess that, but nothing can stop the damage. The entire body is exposed, especially with high oxygen and all that. But without this, the baby cannot survive. So, it is a balance between those. Any other question, please? This is a nice design in the entire body. In fact, if you look at the oxygen tensions in the various parts of the body, right, you know that air goes in through the lungs. So, the lungs have the highest oxygen levels, right, and then it gets distributed to the various parts of the body. So, the cells in the body are not exposed to high oxygen by design itself. For example, if you see here 100% whatever the value, here it is 100 mercury, some measure of oxygen partial pressure. Okay. The red blood cells probably near the lungs it is high, then aorta 80%, arterioles comes down to 80 to 70%, the capillaries is actually only 60 to 70%. Okay. And then so most cells do not see beyond 50 to 60% of air saturation, right. You need to recall your thermodynamics, air saturation, oxygen saturation. We typically in a lot of contexts we talk about 100% air saturation, which means it is saturated with 21% oxygen. So, the theoretical maximum is about 388% of air saturation, because 21% oxygen you multiply it by 488%, you need to multiply it by at least by 5 to get 100% oxygen saturation and so on and so forth. Okay. So, those things we typically deal with air saturation, so be a little careful as to what number you are dealing with. So, most cells, if this is the capillary pressure 50 to 60 mmHg, the cells are seeing less than that because it has to diffuse from here to there and so on and so forth. So, by the very design of the body, your actual cells get to see much less oxygen except the ones that are directly exposed to oxygen or directly receive oxygen, as in the case of lungs and so on. Okay. Any questions so far? Then we will get to specifics from now on, a little more specifics.
How did we develop antioxidant bodies to resist oxygen changes?
Evolution, not us, the initial aerobes. Evolutionary process, you know how natural selection takes place. That is oxygen came in, the cells that were unable to handle oxygen died out. Some of them through gene modifications and so on and so forth by chance, that is what is mentioned, because there is a selection pressure of oxygen here. They developed means by which they could handle oxygen through probably a series of changes. The ones that could handle oxygen survived ultimately, the others died out. Very sad, but nature is brutal. So, that is pretty much what happened to the original aerobes. We did not have to worry, we had that from there. So, any other questions, please? By the way, Sujitra, I need to tell you that reactive oxygen species are heavily involved in progeria. Although the link in the story was only for at the cause at the early stages, the cells of people with progeria have huge reactive oxygen species and so on and so forth. A lot of studies are done, have been done on reactive oxygen species and progeria and so on and so forth. Okay. So, it is not picked only for a story, there is a lot of background to it. Only thing is that the cause wise, it caused DNA damage at some point in time. I just wanted to clarify that. It was not chosen on a whim, it was chosen with basis, but the story part of it is kind of tenuous, that's all. Any other question? Okay. Let us get to some specifics. This gives you an important set of reactive oxygen species reactions in the cell. Of course, this course will be heavily tilted towards reactive oxygen species because we have worked on reactive oxygen species predominantly, very little RNS, if at all any, very little I think. So, that bias will be there. This bias is reflects the bias in the literature also. Most of the work has been done with reactive oxygen species. However, all reactive species could be important, especially reactive nitrogen species and so on and so forth are very important. Only thing is that we have not done much, and therefore, I will certainly mention them. It won't the course won't emphasize them. Do not go away with the idea that they are not important, they are very important. Just that a course, especially elective course, which comes from more from the heart of what we are doing and so on and so forth. So, it will reflect our work, that is natural. Okay. So, this is an important set of reactions in the cell, which happens in every single cell that uses oxygen. A 1 electron reduction of oxygen, this happens in the electron transport chain, it causes superoxide. A 2 electron reduction of oxygen, 1 electron reduction of superoxide gives you hydrogen peroxide. A 3 electron reduction of oxygen, 1 electron reduction of hydrogen peroxide gives you hydroxyl radicals, and a 4 electron reduction of oxygen gives you water. Okay. Remember your electron transport chain, oxygen gets taken in, that is pretty much the only place where oxygen gets used up. Okay. Leave out the minor things. Predominantly the electron transport chain is the place where oxygen is used in the cell, that is the reason why aerobes need oxygen, and that gets converted to water in the process, and this is the electronic way of looking at it. You might want to write this down, I will leave it on for some time. So, oxygen, you know that loss of electrons is oxidation, gain of electrons is reduction. Have we talked about it? We have in the transport course. I did not talk about it in the transport course, right? Okay. Then let me do this. Which one should I? Alt tab, right? Okay, that is, let this be. So, have you heard this? That is a nice mnemonic, we will look at it again. Is it showing up? Yeah. Leo the lion says Ghar. Okay. So, Leo stands for loss of electrons is oxidation, Ghar stands for gain of electrons is reduction. Okay. So, we will be using this, we will look at this again in the appropriate context, I just thought I will mention it here. Okay. So, one you add an electron, gain an electrons is reduction, oxygen plus an electron gives you superoxide, oxygen is being reduced here to give you superoxide. Superoxide gets reduced with, of course, addition of hydrogen is also reduction, both can happen at the same time. You get hydrogen peroxide reduction gives you hydroxyl radicals, and hydroxyl radical reduction gives you water. Okay. So, please make, I hope you have written this down, this will keep haunting you throughout the course pretty much. Okay. Antioxidants, we are just getting a panoramic view of whatever we will look at in this course. Antioxidants certainly are relevant because those are the ones that provide protection to an aerobe against the reactive oxygen species. One of the fates of anaerobes when oxygen levels increased in the atmosphere was to they evolved antioxidant defenses and became aerobes, and the antioxidant is what we are going to talk about now. It is typically understood as a species that delays, blocks or even mitigates oxidative damage to a biomolecule in focus. Okay. This is the currently accepted definition of an antioxidant. This is reasonably complete, so that way it is okay. This can undergo a change as we go along, but as of now an antioxidant is understood, again understood, as a species that delays, blocks or even mitigates oxidative damage to a biomolecule in focus. So, that is what an antioxidant is. You might want to write that down. Just the definition of the antioxidant. Antioxidants can be of various different types. They could be enzymatic. Enzymatic ones are reasonably specific. For example, superoxide dismutase is reasonably specific for superoxide. Peroxidase is specific for hydroxyl. Okay, remember this, peroxidase for hydroxyl. Superoxide dismutase is quite straightforward for superoxide, and so on and so forth. So, you could have specific enzymes against specific reactive species. They could be non-enzymatic, such as ascorbic acid, which are usually not very specific. An antioxidant could be a molecule with a non-specific action, such as glutathione. Okay, you might have heard the term, it is a well known antioxidant. It is nothing but a tripeptide, GSH, first single letter abbreviation of the amino acids, GSH tripeptide, which acts against a variety of reactive species such as hydroxyl hypochlorous acid, peroxynitrite, nitrogen dioxide and others also. Okay, so, it is not very specific, but glutathione is considered an important antioxidant in the cell. A lot of glutathione assays are reported in papers and so on and so forth. Okay. Glutathione levels are reported in papers through assays. There could be proteins that minimize the availability of pro-oxidants such as iron. Okay, iron is an pro-oxidant. Proteins that minimize availability of iron such as transferrin and others. There could be even chaperone proteins that protect the DNA, those are also considered antioxidants, and so on and so forth. Okay, so, expect the definition to become more and more comprehensive with time. At the current state, this gives you an idea of what an antioxidant is. And also strategies such as compartmentalization of molecules such as iron to prevent its reaction with cellular hydrogen peroxide are viewed as antioxidant strategies in the literature. Okay, these are accepted as antioxidant strategies. The iron is kind of limited to a compartment, and therefore, it does not affect the other parts of the cell and so on and so forth. So, it is some kind of an it is considered some kind of an antioxidant action in the cell. Any questions, comments?
So, like ascorbic acid, it is believed to be a source from lemons and oranges.
Yes.
Which is also a rich source of vitamin C. So, can we say that vitamin C is an antioxidant?
Ascorbic acid is vitamin C. A straightforward vitamin C. Another important such compound is alpha tocopherol, which is vitamin E. Okay. So, they are vitamins, they could be vitamins, that is not necessary, not always, but yeah, they could be vitamins. True, yeah, thanks for pointing that out. Any whereas vitamin K3 it is actually menadione which induces reactive species. So, it is not an antioxidant. So, not all vitamins are antioxidants, at the same time some of them, ascorbic acid is certainly a vitamin. which is an antioxidant. Some antioxidants are synthesized in the cell, such as enzymes, proteins, glutathione. Of course, it is a tripeptide, therefore, they are all synthesized in the cells, whereas other oxidants need to be taken through food. Ascorbic acid, alpha tocopherol, these are taken through food. The alpha tocopherol used to come in come as gooey liquid and so on and so forth, which also capsules, certainly. Alpha tocopherol is lipid soluble, it is not water soluble. Ascorbic acid is highly water soluble, just for information. So, that has direct relevance to the way you can give it as a medicine and so on and so forth. Okay. So, the net effect of reactive species generation and oxidant action results in a certain level of reactive species at a particular time point in the cell. Okay. This you are able to relate to, right. You have something that causes reactive species, something that takes away reactive species. Hold that thought, we will develop on that thought as we go along. And reactive species influence many critical functions in the cell, which we are going to cover in some detail in this course. So, at any time there is a certain desired net specific level of intercellular reactive species. They are necessary for the cell. The reactive species, reactive oxygen species especially, and reactive nitrogen species, reactive sulfur species, they are all essential for the cell. Okay. So, at any time there is a certain desired net level of intercellular reactive species, and that desired level results from a balance between the production and consumption rates, right. This is a dynamic system, you can make sense of it only through rates. So, consumption rates of reactive species in the cell, or that is called reactive species homeostasis. Okay. So, the desired level is the homeostatic level. The desired homeostatic level differs with the kind of cell, definitely. The cancer cell typically has a higher homeostatic level of reactive species than a normal cell. In fact, that is what is used to kill it also, as we will see later. The homeostatic level itself of reactive species in a cancer cell is higher than a normal cell, whereas in stem cells they are actually much lower. In fact, stem cells, that is why hide in anaerobic kind of environments in the body, bone marrow and so on and so forth, because they need to maintain a lower homeostatic level than a normal cell. And the desired level could also vary with time. Okay. In fact, there is a nice rhythm in the pseudo steady state level of reactive species in the cell. Okay. And most of you know what a pseudo steady state level is based on our course. If you do not, we will end with that today and then take it up tomorrow, take it up on Thursday. Yeah, I think this is an 8-minute video. So, you watch this, and even if you know, just watch this, it is a refresher for you. Okay. Next slide, that is. In fact, we discovered the rhythms in the reactive species levels in the cell. Before us, it was not known. We discovered it initially in microalgae, chlorella. Ranjani discovered them. In fact, yeah, she discovered them, I should say, between us. Of course, as a group we wrote up the paper, but she discovered them. And then we found the rhythms and pseudo steady state levels in cancer cells, and we expect to find them in all cells. That makes sense. Okay. What is pseudo steady state level? All of you know. What I would like you to do is, we will end the course today with this video. Does any of you, all of you have an engineering background, right? Right. So, you do not need the next video. This video, the ones who have done the transport course already know. For you, it is a revision. Those who have not done the transport course, please look at this. You need to understand this very clearly. Okay. Please listen to this about 9 minutes, and then we will stop today.